All right, welcome back. We're in part 3 here of chapter 3. So facilitate diffusion here. We're still talking about movement of molecules across the membrane here. So it's facilitate diffusion. The process is carried out by specific channel proteins. No ATP is required. You're moving water-soluble molecules across the membrane here, such as amino acids and glucose. And then there's the active membrane transport here, right? So it requires ATP. And again, we're re-highlighting here what this is doing here. You're moving substances against its concentration gradient, right? Allowing them to accumulate on one side of the plasma membrane to the next, right? So the rate of this transport depends upon the concentration of the substrate, the number of pumps that there are, and the amount of ATP or energy you have to transport that. The example of that is sodium-potassium pumps, right? So sodium comes in, potassium goes out. So 3 sodiums come in and 2 potassiums go out. And this is used in muscle cells, right, for contraction, right? And it requires ATP, where you hydrolyze ATP, releasing a phosphate and energy, creating ADP. So there is a thing called secondary active transport. So you're using the potential energy from the concentration gradient, right? So this is kind of like a form. Remember, concentration is high to low, so you're kind of using that as a pump to move one thing across the gradient, right? So one example of this is when you pump out the potassium, like in the primary active transport here, right? Your potassium moves out in this situation. Well, the potassium really wants to move back in, so you can use that mechanism, that action, to pump sodium out, right? And then, of course, once you have sodium back out, then you can use a sodium to, if it wants to come back in, you can pair it with a glucose molecule that wants to move in as well. So you can use the concentration gradients to move other molecules one way or the next based upon the concentration gradient of one molecule trying to move in or out. It's kind of like a dam, in the sense that a hydroelectric dam, you build the dam up, you have lots of water behind the dam, then you slowly release it through, and it spins a turbine, creates energy, releases the water to go down the river. That's kind of like what this is. It's kind of like a pump dam, in the sense that there's a high concentration of something on one side, and it wants to move, say, water or the sodium the other direction. It will use that gradient force to pump something in the other direction. Now, we did talk about and reference vascular transport, so we'll talk about that here. So you're right, this is really used for is you're really transporting very large substances in and out of the cell, right? So we're talking like either parts of a cell, like an immune cell, they will engulf like bacteria, or killer T cells will engulf other cells or bacteria, right? Well, how do they get those large particles in? Endocytosis and exocytosis. So like endocytosis is anything moving into the cell and engulfs it. An amoeba does basically the same thing, but it's prokaryote versus eukaryote. But phagocytosis is where the cell will ingest a solid particle into a large vesicle, right? This is an example of phagocytosis here. This kind of looks like a bacterial cell to me, but they just say particle. Now, penocytosis is an example of where it's actually taking like a big gulp of water, where it's trying to ingest dissolved molecules and maybe even water and brings them into the cell. Same function, where you form like a little plasma membrane bubble but instead of ingesting large particles, you're ingesting like molecules into the cell. So it's like this is like cell drinking is what pinocytosis is. Exocytosis is the opposite. So it's actually moving things outside the cell. Exocytosis is really initiated most of the time by the Golgi apparatus, because that's where all this stuff gets packaged up. There's other vesicles that do exocytosis that are independent of the Golgi apparatus, but that's like an example I have for you of where maybe the cell is just made a hormone and it's trying to release it into the bloodstream. Well, it's going to package it up in the Golgi apparatus and then, in a vesicle and then shoot it out. There is transcytosis where you're moving through the cell where you're endocytosing from one direction and you're exocytosing. I guess I just made it a verb, but go in one direction and out the other, right? It just moves straight through. So it's transient. So it doesn't really stop in the cell or start from the cell. It just moves through. So here's an image of like the pinocytosis and exocytosis, and maybe even, so like here's our cell here. It's an endothelial cell that lines the capillary, right, with a red blood cell here. Pinocytosis, and it drinks it, and the molecules, bloop, like a big gulp, right, and then it excises that out. So this is kind of almost an example of like the transcytosis, right, and then exocytosis that squirts it out on the other side, maybe outside. This is like outside the blood vessel, so it's trying to transport some molecules in, and then of course same way inside. There is receptor-mediated endocytosis. So remember I said there are proteins on the plasma membrane that act as an antennae or receptor receiving a signal. Well, sometimes when these receptors receive a very specific signal, that starts endocytosis. So like a great example of this is like the immune cells, where the killer T cell identifies a bacteria because it's been flagged with the antibody, or maybe identifies the antigen on it and then engulfs it to destroy it, right? So that's a very specific example, because you don't want the immune cells to engulf healthy human cells, so you only want bacteria cells. So you need some sort of way to identify that, and receptors are that way. So that's why a lot of those cells in the immune system utilize receptor-mediated endocytosis. Now, this is just an image of exocytosis here, right? So secretions is really what's going on here. They accumulate in that like plasma membrane bubble here, and then that plasma membrane actually kind of melds into the cell membrane here to release its contents. We have like an actual X-ray imaging, electron imaging, of that, right? An example I have of that is like, again, hormone signaling. So we'll stop the video there. That will end our lengthy discussion on the plasma membrane and transport of molecules in and out and all that fun stuff, which we'll really highlight again in chapter 9 and 10. But we'll stop there.